Hello my dear students, good morning. Welcome you all to a new video of Sunanda's tutorial. So today I am uploading a very special video. I hope it will be helpful for those students who are appearing 12th class board exams. So in this channel from today onwards I will upload details about all your chapters which are present in your biology NCERT biology book. We will discuss each and every page line by line. And today I will discuss the first chapter, that is sexual reproduction in flowering plant. So it is the first page where the importance of flowers are mentioned. Like flowers are objects of aesthetic, ornamental, social, religious and cultural value. They have always been used as a symbol for conveying or expressing love, affection, happiness, grief, mourning, etc. So it is the diagrammatic representation of the longitudinal section or LS of a flower. It is the internal structure of a flower or longitudinal section of the internal or anatomy of a flower. See, in a flower there are 4 holes. The outer two holes are the accessory holes, which are calyx and corolla, and inner 2 essential holes, that is androecium and gynoecium. Individual unit of the calyx is known as sepal. That means the collection of sepals is called as the calyx. Then individual unit of corolla is known as petal. It is the coloring part or most prominent part of the flower. It is the corolla, and the individual unit is known as petal. That means collection of petals is known as corolla. Then 2 reproductive part or 2 essential holes, that is androecium and gynoecium. The individual unit of androecium is known as stamen. Stamens, that means collection of stamens, is known as androecium. Then the female reproductive part, which is gynoecium, the individual unit is known as carpel. So let us discuss about these holes or about this part of a flower in details. First of all, stamen, microsporangium and pollen grain. Figure 2.2 shows the 2 parts of a typical stamen. Stamen means the individual unit of androecium, which is your male reproductive part. Male reproductive part of a flower is known as androecium. Individual unit of androecium or a unit or functional unit of androecium is known as stamen. So this stamen is consist of 2 parts. One long filament with a anther. Stamen consists of a long slender stalk, which is the filament. It is the filament, and terminal bilobed. Bilobed means it have 2 lobes. 2 lobes. Generally bilobed anther. The proximal end of filament is attached to thalamus or petal of flower. The number and length of stamens are variable in flowers of different species. If you collect a stamen each from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissection microscope and making neat diagram would you elucidate the range in shape and attachment of anthers in different flowers. So the size number generally vary in different species of different variety. But the general structure of stamen, it consists of a long slender filament which is very delicate, and terminal anther, which is generally bilobed. A typical anther is bilobed with each lobe having two theca. That means each lobe have two sac in them. That means they are dithecous. Dithecous. It is dithecous. Often a longitudinal groove runs lengthwise separating the theca. Let us understand the various types of tissues and their organization in transverse section of anther, which is present in 2.3 figure 2.3a. So the bilobed nature of anther is very distinct in transverse section of an anther. So the anther is four-sided, that is tetragonal structure. It is a tetragonal structure like this, consisting of four microsporangia located at corners, two in each lobe. Four lobes. The microsporangia develop further and become pollen sacs. They become pollen sacs because pollen grains will be formed inside that sac. They extend longitudinally all through the length of the anther and tagged with pollen grains. Then structure of microsporangium. In a transverse section of a typical microsporangium appears near circular outline. It is generally surrounded by four wall layers. Epidermis. So these are the four layers: epidermis, endothecium, then middle layer, then tapetum. These are the four layers. See, it is the structure of a stamen consisting of this bilobed anther and a long filament. This is anther and this is a long filament. When we cut a section of this anther, we can observe that it consists of two lobes, and within that two lobes, four theca or spore sacs, pollen sacs are present. This pollen sac or overall structure or anther is generally protected by four layers. The outer layer is epidermis, then endothecium, then middle layer, then tapetum. So structure of microsporangium in a transverse section. A typical microsporangium appear near circular outline. It is generally surrounded by four layers: epidermis, endothecium, middle layers, tapetum. The outer three layers perform function of protection. That means epidermis, endothecium, middle layer protect the pollen grains and help in the dehiscence during the release of pollen grain. And the innermost layer, that is tapetum, which nourishes the developing pollen grains. And the cells of tapetum, which provide the nutrition to the growing pollen grain, possess dense cytoplasm and generally have more than one nucleus. That means multinucleate condition. When the anther is young, a group of compactly arranged homogeneous cell called sporogenous tissue occupies the center of each microsporangium. Then microsporogenesis. As the anther develops, the cells of sporogenous tissue undergo meiotic division to form microspore tetrad. So from microspore mother cells, upon meiosis or reductional division, four microspores are formed called as microspore tetrad. When they produce, generally they are present in aggregate manner. They are present togetherly like this and known as microspore tetrad because four microspores are togetherly present. Then as each of the sporogenous tissue is capable of giving rise to microspore tetrad, each one is potential pollen or microspore mother cell. The process of formation of microspores from pollen mother cells through meiosis is called microsporogenesis. So, for the process of formation of microspores from microspore mother cells upon meiosis is known as microsporogenesis. So here are the pollen grains. They are formed from the microspore mother cells upon meiosis or reductional division. The microspores as they are formed are arranged in cluster of 4 cells, so called as microspore tetrad. As the anther mature and dehydrate, the microspores dissociate from each other and develop pollen grains. They separate when they mature, they separate from each other. They detach from that cluster and known as pollen grains. Inside each microsporangium, several thousand microspores or pollen grains are present, and they are formed or released with the dehiscence of anther. And that dehiscence process, generally the endothecium layer helps in the dehiscence of the anther and release of the pollen grains to the outside. Then pollen grain, the structure of pollen grain. The pollen grains represent the male gametophyte or male gamete. If you touch the opened anthers of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grains on your finger. Sprinkle these grains on a drop of water taken on glass slide and observe it under the microscope. You will really amaze that variety of architecture, size, shape, color, design seen on pollen grains from different species. Then the structure of pollen grain. Generally pollen grains are spherical in nature and the diameter varies between different species. It has prominent two-layered wall. The hard outer layer called exine, which is made up of sporopollenin, which is one of the most tough and resistant organic material known. It can withstand high temperature and strong acid and alkali. No enzymes can degrade sporopollenin is so far known. Pollen grain exine has prominent aperture called germ pores, where sporopollenins are absent. Pollen grains are well preserved as fossil because of the presence of sporopollenin, because it is very strong and they cannot be biodegrade easily. Then the inner layer is known as intine. It is thin and continuous layer made up of cellulose and pectin, that means pectocellulosic in nature. The cytoplasm of pollen grain is surrounded by plasma membrane. When pollen grain is mature, it contains two cells. One is vegetative cell, the second one is generative cell. It is the mature structure of one pollen grain. Vegetative cell generally bigger and has abundant fruit reserves and a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. So it is the vegetative cell which is larger and it is the generative cell which is small. Then the vegetative cell has spindle shaped nucleus and dense cytoplasm. In 60% of angiosperms, pollen grains shed at two cell stage. That means dehiscence of pollen grain or release of pollen grain occur at this two cell stage, where only the vegetative and generative cells are present. In the remaining species, the generative cell divides mitotically to give rise to two male gametes before pollen grains are shed. That means in two cell stage. Generally at two cell stage, where the generative and vegetative cells are present, the pollen grain shed or released from the plant or flower. But in some species they shed at three cell stage, where the generative cell divide mitotically to form two male gametes. So in three cell stage, vegetative cell and 2 male gametes are present. 3-cell stage. Pollen grains of many species cause severe allergy and bronchial afflictions in some people, often leading to chronic respiratory disorders such as asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as contaminant with imported wheat has become ubiquitous in occurrence and cause pollen allergy. It is a very important point. Then the pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplement. In Western countries, a large number of pollen products in the form of tablet and syrup available in the market. Pollen consumption has been claimed to increase the performance of athletes and race horses. Then so these are the pollen products which is very rich in nutrients. So it is all about your male reproductive structure of a flower. Then the female reproductive structures, which is pistil, megasporangium or ovule and embryo sac. The gynoecium represent the female reproductive part of the flower. It may consist of a single pistil, which is known as monocarpellary, or they may have more than one pistil known as multicarpellary. When there are more than one pistils present in fused condition, or they attach with each other, known as syncarpous, or they may be free. They may be present in a free condition known as apocarpous condition. Each pistil has 3 parts: the stigma, the style, a long style, and ovary, basal solen portion which is known as ovary. Stigma serves as landing platform for the upcoming pollen grain. The pollen grain, after their release, land on a stigma. Then the style is the elongated slender part as a landing platform, a slender part beneath the stigma. So beneath the stigma a long slender style is present. Then the basal part, which is ovary. Inside the ovary, ovarian cavity or locules are present like this. The placenta is located inside the ovarian cavity. Arising from the placenta, megasporangia is present, commonly known as ovule. The number of ovules in each ovary may be 1, as seen in case of wheat, paddy and mango, to many, as seen in case of papaya, watermelon, orchids. They have many ovules inside the ovary. So suppose it is the ovary. Inside the ovary many sacs or rooms are present, and inside that sac many ovules are present. Many or one ovules are present inside that ovary. Ovules are present, and the number of ovules vary. It may be 1 or it may be many. Then the ovule. Let us familiarize ourselves with the structure of a typical angiospermic ovule. The ovule is a small structure attached to the placenta. By means of a stalk called funicle. So it is the structure of ovule, and it is attached with the attached to the placenta by means of stalk called funicle, and the body of ovule fused with funicle in the region called hilum. The place where the ovule attach with the placenta is known as the funicle. The ovule attached with the placenta by means of a stalk called funicle. The body of the ovule fuse with funicle at the region called hilum. Thus hilum represents it is the place, that is hilum. Hilum represent the junction between ovule and funicle. Each ovule has one and or two protective envelope called integuments. So these layers are integuments. Then integuments encircles the nucellus except the tip where a small opening is present. So these integuments totally cover nucellus. At some places nucellus is absent. Here nucellus is absent, and that opening is called micropylar opening. Opposite to micropylar end, where micropyle is present, that end is known as micropylar end, and opposite to that chalazal end is present, representing the basal part of the ovule. Enclosed within the integuments, a mass of cell called nucellus or parenchymatous tissue are present. Cells of nucellus have abundant reserve food material. Located in nucellus is the embryo sac. Inside the nucellus, embryo sac or female gametophyte is present. An ovule generally has single embryo sac formed from the megaspore. Then the process of megasporogenesis. The process of formation of megaspore from the megaspore mother cell is known as megasporogenesis. Ovule generally differentiate a single megaspore mother cell in micropylar region of the nucellus. It is a large cell containing large dense cytoplasm and a prominent nucleus. Megaspore mother cell undergoes meiosis. Then after meiosis, as the result of meiosis, megaspores are present. Then female gametophyte. In majority of flowering plant, one of the megaspore, see, when from megaspore mother cell, 4 megaspores are formed. Generally, one megaspore remain viable or remain active, and other 3 are degenerated. So that active or remaining functional megaspore will divide further. Only the functional megaspore develop into female gametophyte or embryo sac. This method of embryo sac formation from a single megaspore is known as monosporic development of female gametophyte. Then let us study the formation of embryo sac in a little more details. The nucleus of functional megaspore divide mitotically to form 2 nuclei. At first, it will form 2 nuclei. 2 more sequential mitotic nuclear division results in the formation of 4 nuclei. Later 8 nucleate stages appear. It is of interest to note that these mitotic divisions are strictly free nuclear. That is nuclear division that are not followed by immediate cell division. Freely they divide without any cell wall formation. After 8 nucleate stages, cell wall are laid down, leading the organization of a typical female gametophyte or embryo sac. Observe the distribution of cell inside the embryo sac in the figure number 2.8. So it is the fully organized female gametophyte or mature embryo sac where 8 nuclei are arranged in chalazal end. In the chalazal end, 3 cells are present which are known as antipodal cell or antipodals. In the micropylar end, 2 synergids with a egg cell are present. In the middle, 2 nuclei are present in a large central cell. So a typical mature embryo sac have 8 nuclei and 7 cells, because in the middle 2 nuclei are present, but it is a single cell, and inside it 2 nuclei are present. So overall a mature embryo sac is 7 celled and 8 nucleate. So it is all about your male reproductive organ and female reproductive organs and their development. In my next class I will discuss about the pollination, fertilization, and others present in this chapter. So stay tuned and enjoy my videos like this. It is the part 1 video. I will upload part 2 video of this chapter very shortly. So stay tuned and most importantly subscribe my channel to get more videos like this. Thank you.